Hi. I want to talk to you about TRIF, the new biological automatic detergent. It's a revolution for wash day. We took an ordinary shirt, tea time stains, those soiled neckbands, and those personal stains we don't like to talk about. Well, we gave it the TRIF treatment. And I think
Yet another miracle biological powder. Just what we need. But what is all that biological stuff anyway? And how is it different from non-biological powders? Meet the A-team. Their assignment, if they choose to accept it, is to turn starch into sugar in the shortest possible time. The starch molecule looks quite complicated until you realize that it's just large numbers of sugar molecules linked together. Break the links between the sugar molecules and the starch turns to sugar. Easy when you know how. So how would you do it? Andy decided to heat the starch solution to 37 degrees centigrade. Anne's going to try boiling it, and Algernon, who is the brains of the outfit, has decided to use a chemical substance to make the reaction go faster. He chose a non-biological reagent, hydrochloric acid. Andy has been warming the starch at 37 degrees centigrade for about half an hour. That tile already has a drop of iodine indicator. Sugar will turn it colourless. Starch will show up dark blue. The blue-black colour means that Andy's experiment still has a long way to go. In fact, if we look at the projected time for the starch to be converted into sugar by this method, you can see that he won't be finished until this time next week. In comparison, Anne's doing pretty well. By adding more heat energy and boiling the starch, she's managed to break down more of the starch than Andy has. Algernon has the best results so far. At this rate, the starch will turn to sugar in about a day. Still not very good, though. Does anybody have a better idea? This looks like the same experiment we saw previously. The water bath, like Andy's, is at 37 degrees centigrade, and the experiment has been running for exactly 2 minutes. The colour of the iodine indicator remains unchanged. The starch has been broken down to sugar extremely rapidly by an enzyme, amylase. The amylase has speeded up the reaction tremendously. It's many times faster than the best A-team result, which combined heat energy with a non-biological reagent. We've seen that heat energy on its own can help to speed up a reaction. So, can we speed up the amylase reaction even more by increasing the temperature from 37 degrees centigrade to boiling point? Nice try. Shame about the results. Enzymes are damaged and then destroyed at temperatures above 40 degrees centigrade. Many of them work best at 37 degrees centigrade. Let's look at the figures again. Raising the temperature of the starch solution to boiling point and keeping it there for many hours used up large amounts of energy. The lower temperature at which the enzyme reaction took place and its far greater speed meant that it used up far less energy. Enzymes reduce the amount of energy needed to make a reaction go, which is why TRIF cleans clothes at lower temperatures. These food stains contain starch and protein. The enzymes in TRIF rapidly break them down to smaller molecules that dissolve and can be washed away.
So, when it comes to removing those stubborn stains, You could try boiling them away, or you could add an acid reagent. But so much simpler and more efficient, just to add a pinch of enzyme. And there's the proof. Biological powders really can wash cleaner. It'll save you heating bills, and you won't lose your shirt either. Use a biological catalyst. Now that's efficient.
So where do these amazing biological catalysts come from? They're formed inside living cells like these, where they are built up by assembling small subunits called amino acids into much larger molecules called proteins. Each protein has its own unique sequence of amino acids. These long chains then fold up, usually into a spiral, but the proteins, which are going to be enzymes, fold up again into more complex shapes. The exact shape is very important, because when an enzyme such as amylase acts on a substrate such as starch, the enzyme and substrate must fit together like a lock and key. Here, an enzyme is breaking down a blue substrate molecule into smaller products. This is called a catabolic reaction. You should by now be able to predict some of the key properties of enzymes. Key property number 1. Because the enzyme is not used up in the reaction with the substrate, a small amount of enzymes can go a long way. 2. Because the fit of the lock and key is so important, enzymes are very specific and will act on only one type of substrate molecule. 3. Anything that changes the shape of an enzyme, and hence its ability to fit its substrate, will make it less effective. Boiling, for example, terminates all enzyme activity. So when you think of enzymes, think of the efficiency of enzyme reactions and their high specificity, which is why all living organisms rely on large numbers of different enzymes. Enzymes speed up chemical reactions, which would otherwise take days or years to occur, or which could only happen at high temperatures. They regulate all of the different chemical reactions which make up our metabolism and which make life possible. Without enzymes, for example, the humble potato would be completely inedible. The molecules of vegetable starch are too large to be absorbed. In the gut, starch must be broken down by digestive enzymes into smaller glucose molecules. These can be easily absorbed and then transported in the blood to the liver and muscles. Here, the glucose molecules are joined together by a second group of enzymes to create animal starch, or glycogen, which is the form in which animals store carbohydrate. Then, when the muscles are being exercised, the glycogen is broken down by a third group of enzymes to glucose once more to release its stored energy. By now, you can see that the simplest activity, like sitting down and eating a packet of chips, is actually far more complicated than it looks. Enzymes are involved in breaking down the vegetable starch in this vegetable to glucose, and then building it back up again to animal starch in this young animal. Enzymes that break down large molecules, like starch, into smaller subunits such as glucose are called catabolic enzymes. Enzymes that build up smaller units into larger ones are called anabolic enzymes. The A team used the catabolic enzyme amylase to break down starch into sugar. Potatoes contain amylase, And if we blend a potato and filter and centrifuge the result, the potato's own starch is left behind, and we are left with an extract rich in amylase. Take a Petri dish with agar, already containing sugar and iodine reagent. What will happen if we add the amylase extract? The sugars slowly build up into starch. But how can amylase be anabolic as well as catabolic? If there is a lot of substrate, say glucose, and not much product, starch, the reaction will go in the direction of substrate to product. If the product is beginning to build up and there's not much substrate about, the reaction will tend to go into reverse. By removing product, which is what usually happens in nature, the reaction is encouraged to go from substrate to product, whether it's an anabolic or a catabolic reaction. Humans have been using these properties of enzymes for thousands of years. Yeast contains oxidizing enzymes that act on the substrate, sugar, to produce various products. By starting with large amounts of sugar, the reaction can be pushed in the direction of product formation. If oxygen levels are reduced, by sealing the reaction vessel, the main product is alcohol, with as byproduct large amounts of carbon dioxide gas. The baker relies on yeast, just as the brewer does. But here, the carbon dioxide isn't a byproduct. It's an essential part of bread making. After the dough has been mixed, it is set aside to rise. As the yeast enzymes oxidize the sugar to carbon dioxide, bubbles of the gas lift the dough. What happens to all the alcohol? How would you design a biotech plant to produce high-quality protein from cheap and widely available materials? The plant should also be self-repairing, low on maintenance, mobile, and self-replicating. Give up. The cow is, amongst other things, an amazingly efficient bioreactor. It converts grass into milk, meat, and so much methane that cows are now making a major contribution to the greenhouse effect. So how do they do it? Well, the cow uses enzymes, but they're not the cow's enzymes. The grass that the cow eats is broken down by enzymes produced by microorganisms that live inside the cow's stomach, where they form an essential part of its metabolism. We're now inside the stomach of a live cow. There's its digestive juices flowing over its breakfast of chopped maize, which it's only just finished eating. As the chopped maize sinks into the stomach, you can see the inside of the stomach wall and the beating of the cow's heart. And if you listen, you can hear the gas released by the enzymes breaking down the substrate as cellulose is turned into cow. We'll place a small sample of the stomach contents under the microscope. The tiny particles are bacteria. Their enzymes break down cellulose to fatty acids. The large organisms swimming across the screen are ciliates, and they eat the bacteria. The bacteria and the ciliates are then digested by the cow. Industry has learned how to build mechanical cows of a sort. A bioreactor is basically a vessel full of microorganisms. Add substrate, such as vegetable waste, stabilize the temperature and oxygen levels so that the microorganisms can multiply, and then harvest them. They're usually used as a source of enzymes, but they also produce other valuable substances which are made into antibiotics and other products. Typical microorganisms include these yeasts, which provide many enzymes, including protease, and this fungus, Aspergillus, a rich source of amylase. Once the microorganisms have been harvested, the enzymes and other biological products are collected and purified in production plants like this, in batches of many thousands of gallons at a time. The whole plant is computerized. Each step of the process is rigorously monitored and controlled to prevent contamination. So, how are the biological products extracted? Well, first, the fluid medium containing the enzymes, or other proteins, is treated to make the proteins insoluble. They come out of solution. Then the liquid is removed by ultracentrifugation in these refrigerated centrifuges. It's a complex procedure. Why do you think that refrigeration should be necessary? Finally, here is the semi-purified extract. What are these industrially derived enzymes used for? The food industry uses large amounts of sugar and produces a lot of waste starch. Rather than throw the starch away, they use the enzyme amylase to convert it into sugar. That sugar is used in jam, in soft drinks, and in less obvious foods such as ham, salad cream, tomato soup. Even in canned vegetables. What effect do you think this must be having on our health? This operation is to repair the damage done by a heart attack when a blood vessel supplying the heart was blocked by a blood clot. In future, a bacterial enzyme, streptokinase, may replace surgery. An injection of streptokinase dissolves blood clots quickly and selectively. There is a lot of talk about environmental pollution, but before we can discuss it meaningfully, we have to be able to measure it. How would you detect small amounts of pollutants in the water supply? Well, enzymes can detect very low levels of the substrate they are specific for. Enzymes used in this way are called biosensors, and they begin in the bacteriology lab. These are cultures of a bacterium called Syncyticococcus. Green because they contain chlorophyll and depend on photosynthesis. A drop of the bacterial culture is placed carefully onto a disc of porous plastic. A disc of the same material is laid on top of it, and the bacterial sandwich is placed on a metal electrode, fastened in place with nylon gauze, secured, and the electrode is now ready for testing. Light is shone on the electrode, and enzymes in the photosynthetic bacteria respond by altering the electric charge of the microorganisms. This is a sign of their good health. But add a pollutant, and the bacterial enzymes are poisoned. The electrical response to light falls. These biosensors are so sensitive they can detect pollutants at concentrations as low as 20 parts per billion. Which pollutants do you think these bacteria are so sensitive to?